So final chapter, chapter 18, we're going to be talking about cancer. So a quick overview on cancer if you click the link below. So here are just a few quick facts. Did you know there are over 100 different types of cancer? And 90% of cancer deaths are due to metastasis. So that's just spreading of the cancer to different locations in the body. And so cancer cells, they usually leave the tumor and spread throughout the body. Only a small fraction survive. And also cancer cells need oxygen and other nutrients to grow. So cancer cells will send the signals to the blood vessels to start angiogenesis, which is formation of the new blood vessels. So here's just the estimated cancer deaths in 2009. So 30% was lung cancer in males, 26% lung cancer in females, 9% prostate in males, 15% breast cancer in females. So you'll notice that lung cancer is the leading cause of cancer deaths in both males and females. So here are your learning objectives. Know them well. I heard that he didn't finish all of the slides, so you guys are not responsible for learning objective 11, 12, or 13. So here we have a lot of terms relating to cancer. This is your first learning objective, so know this slide very well. So the first term we have is benign, which is just a harmless tumor. So a common example is a wart. Malignant means that it now is dangerous and it can rapidly grow and begin to destroy your surrounding tissue. Metastasis is where those cells will establish new growths into different locations in the body. A proto-oncogene is just a normal gene that regulates cell growth. But an oncogene is a mutated proto-oncogene, so now it has the potential to become something dangerous. And then we have tumor-suppressor genes, which are, a lot of drugs have these. They suppress the growth of cancer cells by inhibiting proliferation, promoting cell death, and repairing DNA. So here's just a visual representation of how cancer develops. So let's say we have normal cells, and these normal cells are called proto-oncogenes. Now, if we have a mutation in the proto-oncogene or the tumor suppressor gene, so the one in orange, that is going to begin to grow. So if it begins to grow and proliferate, that's called malignant. So once you have multiple mutations in the proto-oncogenes, mutations in the tumor suppressor genes, then it's going to invade and surround the tissue. And then if it invades a blood vessel, that's called metastasis because now it's expanding to different areas in the body. Learning objective 2 is to know the damage to DNA that leads to mutations. So number 1 is chemical and physical alterations in DNA. So these mutations can be caused by nitrosamines, which are found in some of the foods we eat. So you don't need to know the mechanism of it, you just need to know that a mutation in DNA can be caused by nitrosamines, and that's an example of chemical and physical alterations. So nitrosamines are sometimes found in foods like fish, and they're usually responsible for high incidence of gastric cancer. So other chemical and physical alterations in DNA include mutations caused by UV, mutations caused by benzo-A-pyrene, which is from cigarettes, and we've learned about that before. So here's a slide we've seen before, oxidation of benzo-A-pyrene. So benzo-A-pyrene is not carcinogenic until it's oxidized within the cells. It covalently binds to guanine, and that will interrupt hydrogen bonding because it distorts the helix. So a smoker is going to oxidize their benzo-A-pyrene inside their cells. The benzo-A-pyrene will covalently bond to guanine, therefore messing up the hydrogen bonding between guanine and cytosine. So this distorts the helix, causing a mutation. So the next damage to DNA leading to mutations is something called gain-of-function mutation in proto-oncogenes. So if you remember from your definitions, proto-oncogenes are just a normal gene that regulates cell growth, and it can be dangerous if it's converted into an oncogene, and this is usually caused by a gain in function in the DNA. So here we have a proto-oncogene, and let's say radiation or some chemical carcinogen affects it. So that is going to cause a mutation in the coding region, which it will produce a hyperactive protein. So that means there's going to be an increase in the rate of production of the proto-oncogene, and with increasing rate of production, there's more room for error. So that's usually what causes a mutation. So the other damages to DNA that can lead to mutations are absence of the repair enzymes and then mutations in the repair enzymes. So be able to tell me which 4 things can lead to a damage to DNA, and then know the examples of each. So the first one we talked about mutations caused by nitrosamines, mutations caused by UV, and mutations caused by benzo-A-pyrene. Then we talked about the gain of function mutation, where radiation can have a adverse effect to the DNA sequence. And then we have absence of repair enzymes and mutations in the repair enzymes, and the following 3 examples. So in the absence, or if there's a mutated endonuclease, then there won't be anything to remove the damaged or distorted region of the DNA. So this is what's going to lead to mutation. So the key thing is really just knowing that in the absence of your endonucleases, or if there's a mutation in your endonucleases, then there won't be anything to remove the damaged or distorted region of your DNA. And this is what leads to cancer. So DNA mismatch repair is a system to recognize and repair erroneous insertions, deletions, or misincorporations of bases. So anything damaging to your DNA. So if we have a mutation in the mismatch repair enzyme, we're not able to repair the system. And that's another cause that can lead to cancer. So here's learning objective 3, knowing the classes of oncogenes and the mechanisms of activation. So this is a very important slide to remember. He could easily ask you a question like, cell cycle regulators have the following activation mechanisms. So knowing that it's translocation, amplification, and point mutation would get you the right answer. So be able to differentiate between all of these. So here's a more in-detailed table of what we just saw on the previous slide. So definitely look over this table, especially the ones he underlined. For example, if we are talking about apoptosis regulators, the proto-oncogene for that apoptosis regulator is BCL2, and its activation mechanism is translocation. So being able to relate all of these things together is necessary for that learning objective. So learning objective 4 is knowing the role of proto-oncogenes and oncogenes in signal transduction and cell cycle progression. So oncogenes in the signal transduction cascades, they're growth factors and growth factor receptors, signal transduction proteins, transcription factors, and we'll be talking about those in the next few slides. So here we have a signal cascade, which you guys seen before. He's not going to test you guys again on the signal cascade, but he just wants you to know that if you have too much growth factor or growth factor receptor, then your target cell might respond by proliferating inappropriately. So this is what could lead to cancer, the rapid growth of something. So here we have another signal transduction pathway. So RAS, if you remember, activates the MAP kinase pathway. And if that pathway is active, it's going to stimulate transcription. So over here it's showing uncontrolled cell proliferation. So if RAS stays active for too long, then it's going to activate the proto-oncogene transcription factors Mike, Phos, and June, which is going to increase the rate of transcription. So again, if you increase the rate of something, then there's more likely of errors to be produced, and that's what's going to lead to cancer. So next we'll talk about oncogenes in the cell cycle. So before we talk about how it affects cancer, we have to understand the cell cycle. So cyclin or cyclin-dependent kinases control the progression from one phase of the cycle to another. So here's just showing how CKI, which is your cyclin-dependent kinase inhibitor, can inhibit the cyclin and CDK activity. So at the top we have an active complex, cyclin and CDK, but if CKI comes into the picture, then it makes it an inactive complex. So now that we have an idea of cyclin synthesis and CKI inhibition in the cell cycle, we can talk about the control of G1 to the S transition. Alright, so this is how we control the G1 to S transition in the cell cycle. So let's say we have a growth factor. Growth factor binds to the receptor and then produces a signal cascade. And let's say it activates the RAP pathway. So that's going to increase or stimulate transcription. So if you remember from the previous slides, if a cyclin binds to a CDK, we have an active complex. So in this case, we have CDK4 and CDK6. Cyclin D binds to both of them, and now you have two activated CDK complexes. On the bottom half, we have a transcription factor called E2F, and then we have RB, which is retinoblastoma protein. So normally, if these two are complex together, then it's an inhibited complex. But once cyclin D comes into the picture, it's going to phosphorylate your RB, and that's going to cause it to free itself from E2F. So E2F can now bind to DNA and increase gene transcription. Also pay attention to CKI. Remember, CKI is your cyclin-dependent kinase inhibitor. So if that binds to your activated CDK complex, that will inhibit the complex from forming. So learning objective 5 is describing the role of various tumor suppressor genes. So here's a very important table to remember, especially the ones that are in color. So you have the class, signal transduction. You have the protein, NF1. You have the location, and then the associated diseases. So definitely know these. So tumor suppressor genes that regulate cell cycle directly is the retinoblastoma gene, which we just talked about. So this is just showing you what happens if the RB gene is mutated. So normally, if an individual inherits a mutated copy of the RB allele, like in picture B, there's a 100% chance that that individual is going to develop retinoblastoma. So that's called familial retinoblastoma. But if you look at picture A, they only have a 50% of developing it because they have a normal functional RB gene. So if the other one gets infected, they're still okay, but if both get infected, it's called sporadic retinoblastoma. So another tumor suppressor gene is called P53, and that's also known as the guardian of the genome. So P53 is the guardian of the genome. If it encounters a DNA that is damaged, your P53 levels are going to rise. And P53 can do a couple of things. So it can stimulate the transcription of P21 gene product. So that P21 gene product is going to inhibit the cyclin CDK complex. And if the cyclin CDK complex is inhibited, then there's nothing that can phosphorylate the RB gene. And if nothing phosphorylates that RB gene, then you can't release the E2F protein, which is needed for transcription. So you prevent the cell from entering the S phase. So P53 can also do another thing. It can stimulate the transcription of GAD45, which is a DNA repair enzyme. So GAD45 is going to try to repair the damaged DNA. And the last thing it can do is it can activate two genes called the BAX gene and the IGF-BP3 gene. And IGF-BP3 inhibits the anti-apoptotic signals so that the BAX gene can activate apoptosis and kill the cells that are damaged. So another tumor suppressor gene that affects receptors and signal transduction is the regulators of RAS. So go back a few slides to the table. NF1 appeared in the table, so definitely know NF1 is associated with RAS, and its function is basically to decrease cell proliferation. So if you look in the picture, RAS has GTPase activity, and NF1 is going to activate its GTPase activity, thereby decreasing cell proliferation. So it makes RAS inactive. So this slide is just FYI. So the next tumor suppressor genes talk about patched and smoothened, and then tumor suppressor genes that affect cell adhesion. So here we talk about catenins and cadherins, and cadherins form complexes that bind cells together. So if you have the loss of E-cadherin, then you're more likely to have the cancer cells detach and migrate in metastasis. So individuals who inherit a mutation in E-cadherin are predisposed to developing some sort of cancer, especially gastric cancer. So here we talk about two important things, APC and beta-catenin. So beta-catenin can act as a transcription factor, and it can travel to the nucleus where it increases synthesis of MYC and other genes that stimulate cell proliferation. So APC is a tumor suppressor gene that normally binds to beta-catenin, so it can't do that. So let's say we have a mutation in APC, that's going to cause APC to become inactive. So if APC becomes inactive, beta-catenin can travel to the nucleus and stimulate that cell proliferation. So if we have increased stimulation of transcription, then that's what's going to lead to more errors and possibly cancer. So here's just a summary of Learning Objective 5 describing the role of various tumor suppressor genes. So now we talk about apoptosis. This is learning objective number 6, so knowing the normal pathways to apoptosis and how cancer cells can bypass it. So apoptosis is programmed cell death in normal cells. There's 3 phases: initiation phase, a signal integration phase, and an execution phase. So here's just an overview of the signal transduction pathways that involve apoptosis. So P53, remember, activates the Bax gene, which stimulates apoptosis. We talked about P21, which inhibits that Cyclin CDK complex. We talked about RB and the E2F. We talked about APC and beta-catenin, and we'll talk about the BCL2 gene and the capsases in a few slides coming up. And we also talked about RAS. So one of the pathways to apoptosis are caspases, which are proteases that cleave peptide bonds and initiate a signal transduction pathway that leads to apoptosis. So in the previous slide, we talked about caspases briefly. There's 2 pathways that can lead to apoptosis using caspases. One is the death-receptor pathway, and the other is the mitochondrial integrity pathway. So we're going to talk about both of these in a few slides coming up. So the first pathway we're talking about is the death-receptor pathway, and I have a video under my chapter 18 links if you want to watch that. So here we have a death-receptor pathway. Once a ligand binds to the death receptor, it can activate an adaptor protein FAD, which stimulates autocatalysis of procaspase 8 and 10. So 8 and 10 become active caspases 8 and 10, which can activate procaspase 3, 6, and 7 into execution caspases 3, 6, and 7. So once you have these active execution caspases, they can begin going through chromatin condensation, DNA fragmentation, or surface alterations. And execution caspase 3 can also cleave BCL2 to form BID, and BID will stimulate the activation of the mitochondrial integrity pathway, which is the other pathway that initiates apoptosis. So you should know which caspases do what. So 3, 6, and 7 can go through chromatin condensation, DNA fragmentation, surface alterations, but caspase 3 can also cleave BCL2, which activates BID to form and activate the mitochondrial integrity pathway. So the other pathway that initiates apoptosis is called the mitochondrial integrity pathway. And a key step to this is having cytochrome C. So we're going to talk about this in the next slide. So here we have the mitochondrial integrity pathway. So mitochondria, once it gets a death signal, will release cytochrome C. And cytochrome C will bind to APAF1. And along with ATP, they're going to form something called a apoptosome. So the apoptosome converts procaspase 9 to active caspase 9, which initiates the apoptosis pathway. So the last thing we'll talk about is the BCL2 family, and these are the decision makers. So they decide if a cell is going to die or not. So here are some of the roles of the BCL2 family members in regulating apoptosis. So BID can go one of 2 ways. It can follow the anti-apoptotic route. So that's BCL2, which is anti-apoptotic, so your cell will not die if it goes this route. or it can follow the pro-apoptotic route, which includes the protein Bax. So if it goes the pro-apoptotic route, then Bax is going to release the cytochrome C from the mitochondria. So this would be the mitochondria integrity pathway. And once cytochrome C is released, we know it complexes with APAF to form the apoptosome to activate caspase 9. So this initiates apoptosis. So here are a few factors that can stimulate apoptosis. So withdrawal of growth factors, elevation of p53, monitoring of DNA damage by repair enzymes, and release of TNF, which is tumor necrosis factor or other immune factors. And then also pay attention to the bottom red. This was actually an exam question. So mutations in the oncogenes can create apoptosis-resistant cells. So in the previous slide, we talked about how mutations in oncogenes can create apoptosis-resistant cells. So that would mean it would go the anti-apoptotic route. So know this family of BCL2. Anti-apoptotic would include BCL2, X, and W. Pro-apoptotic would include Bax, BAC, and BOC. And then pro-apoptotic BH3 is BAD, BID, and BIM. So this slide is just FYI. So luckily, cancer requires multiple mutations to occur. Here are the possible steps in the development of colon cancer. So just read through a few of these and know that cancer requires multiple mutations to occur. So learning objective 9 is defining the mechanism of rapamycin. And rapamycin is a drug that inhibits the activation of P70S6 kinase. It inhibits the activation of CDK2 cyclin E complex. It inhibits the phosphorylation of retinoblastoma protein. And it inhibits the suppression of CDC2 and cyclin A transcription. So those would be good things to know that rapamycin inhibits. And so it prevents this from occurring by interacting with this mammalian target of rapamycin called mTOR. So interacting with mTOR prevents it from phosphorylating the S6K kinase. So this is just the mTOR signaling pathway with rapamycin. He won't test you on this, so just know the previous slide and you'll be good. So here we're just introducing a drug called busulfan to a person who might have symptoms for CML, which stands for chronic myelogenous leukemia. So alkylating agents like cyclophosphamide have been used alone or in combination with busulfan. So here are the two drugs we just talked about, busulfan and cyclophosphamide. You don't have to know the details of the drugs, just know that busulfan and cyclophosphamide can be used to treat CML. So purine and pyrimidine antagonists and hydroxyurea are also sometimes effective in CML. So purine antagonists function by inhibiting DNA synthesis in two ways. So the first way is just inhibiting production of the purines adenine and guanine. So if the cell doesn't have enough purines, then we're not going to be able to base pair. So DNA synthesis will be halted and the cell cannot divide. Another way is they have anti-metabolites that they can incorporate into the DNA molecule during synthesis, and this is going to interfere with cell division. And then for pyrimidines, they can block the synthesis of the nucleotides C and T in DNA, or C and U in RNA. So the drugs used to block the construction of these nucleotides have structures that are similar to the natural compound. So here we have a closer look at the purine antimetabolites. One of the best known purine antimetabolites is acyclovir. So let's say a patient has a herpes virus infection. We can give them acyclovir and they have purine antimetabolites that look almost identical to the original purines. So 6-MP is the antimetabolite of adenine and 6-TG is the antimetabolite of guanine. So they act as false substitutions in the production of nucleic acid, so it's going to inhibit nucleic acid synthesis. So here's an example of a pyrimidine antagonist, 5-fluorouracil, or 5-FU. It blocks DNA synthesis by blocking the function of uracil, and it can also induce cell death by blocking transcription and inhibit cell growth. So I would know the example of pyrimidine antagonists. So 5-FU is a pyrimidine antagonist, and then also know the purine antagonist, which would be 6-MP and 6-TG from the previous slide. So here's a pathway that can stimulate the growth of cancer cells. So in chemotherapy, we're going to target certain enzymes in this pathway to hopefully kill off the cancer cells. So methotrexate and trimethoprim are going to inhibit dihydrofolate reductase, which means that you can't recycle the N5-N10 methylene H4 folate. So this will interrupt the cycle. And then you also see FD-UMP, which is an antimetabolite of 5-fluorouracil, can inhibit the thymidylate synthase. So here again, same drugs that we saw in the previous slide. Fluorouracil is converted to FD-UMP, which inhibits thymidylate synthase, and then trimethoprim and methotrexate inhibit dihydrofolate reductase. So know which drugs inhibit which process. So the last one we'll talk about is hydroxyurea to treat chronic myelogenous leukemia. So hydroxyurea, or INN, was an anti-cancer drug synthesized in 1869, and it's an inhibitor of the enzyme ribonucleotide reductase. So ribonucleotide reductase converts ribonucleotides to deoxyribonucleotides for DNA. So if it inhibits that, then it can't convert the ribonucleotides to deoxyribonucleotides, and therefore you stop the synthesis of DNA. So there have also been trials with gamma and beta interferons that have shown promising increasing survival in CML patients. So interferons are proteins that are released by your cell in response to the entry of a virus. So they don't kill cancer cells, but they can reduce the growth of cancer cells by regulating the action of several genes that control secretion of the cellular proteins that affect growth. So Gleevec was a drug developed to treat CML. In people with CML, they have this protein called BCR-ABL, which is found only in cells expressing the Philadelphia chromosome and not in normal cells. So Gleevec was specifically designed to block BCR-ABL function. So that would stop cell proliferation, and in some cells, they would induce apoptosis, so the cell would die. And since the normal person doesn't have the BCR-ABL gene, then the drug Gleevec wouldn't affect a normal person. So this has been very promising, but there has been some problems. Some patients suffer relapses, and some people who take Gleevec actually have an amplification of the BCR-ABL gene. Okay, so I'm going to stop it here because Dr. Schroeder said that objectives 11, 12, and 13 were not going to be tested on.